hit that head on. So again, welcome everyone. We're really excited that you're all here today. This is going to be a roundtable discussion on implementing machine learning and biomolecular research and limitations and opportunities. We have Elucidata, Altus Labs, Atomwise, and Cyclica today, and 2015 Visionaries will be moderating as best as I can. Today's agenda, brief industry perspective, and then I'll do the speaker introductions, and then I will do we'll do the roundtable, and during that roundtable, I will ask questions, the audience will ask questions, and it should work out really well. Rules of engagement, put your questions in the chat or Q&A box. The Q&A box is set up for that, but sometimes people are more comfortable with the chat box, so whatever works best for you. I'll moderate questions with the panelists. The panelists can ask questions and answer questions, so it's really about somewhat controlling but not over controlling the situation, and we just really want to get a lot of good substance, a lot of good knowledge sharing out. Based on the poll that we just saw, there's a lot of people that could be looking for advice or help in the near future, so it was good to see that. And of course, this will go on demand afterwards, and you'll get to see the questions and the poll results and all that stuff, so that's good. So I'm going to give an industry perspective. I'll share with you a thought, and people can deny or say you're crazy or I agree or disagree, whatever. But I see machine learning a little bit like advanced mimicry, where we're all looking for model quality data and we're trying to reproduce something that we've observed and be able to predict or suggest, and this is a really important part, suggest a path forward. And sometimes when you over predict or prediction becomes an absolute, then sometimes that can stifle intuition and innovation. So you have to be really careful how you approach it in the in industry. So there's the types of machine learning or artificial intelligence. We're not going to talk about linear algebra or anything like that. So we've got artificial intelligence, and that's the programs with the ability to learn and reason like humans. It's gotten a lot of press in the past 2 weeks with some of the things brought out by Google, which was, in my opinion, very interesting, but not surprising. Machine learning, algorithms with the ability to learn without being explicitly programmed. And then deep learning, a subset of machine learning in which artificial neural networks adapt and learn from vast amounts of data. And of course, this is just a very high level approach, and you're going to hear some expertise today from the panel. So some terms and definitions. Supervised learning requires training labeled data, neural nets, k-nearest neighbors, Bayesian, etc. Unsupervised learning does not require labeling data explicitly, clustering, k-means, PCA, and then ROC curve, a graphical representation of the contrast between true positive rates and the false positive rate at various thresholds. Some more definitions. Bias, error due to artificial or overly simplistic assumptions in the learning algorithm you're using. This can lead to low predictive accuracy or an invalid model, to be fair. And then variance is error due to high complexity in your algorithm, which can lead to overfitting or training data. Again, your model will fail in its test set and applicability. One thing I thought it was important to touch on is hype, and we've all dealt with it. I think we've dealt with it from the late 90s when it comes from computational chemistry and machine learning, etc. And in my opinion, there's two experiences that I've seen. Industry consulting organizations, and I know we're a consulting organization, but we try not to do this, push premature technology in order to drive future strategy discussions, which unfortunately can lead to a lot of disruption of innovation in a company. I've experienced that firsthand working in large biopharmaceutical companies. Then the second part of it is the corollary in a way is the pressure from the hype drives organizations to raise their game. So I have seen both of these, and this can come out in discussion today as well. It can be detrimental, it also could, potentially be positive. So today I would like to introduce Shefali, Henry, Felix, and Ali, and I'm going to do that. Can you guys still see the slides? Just want to make sure. Yeah, okay. So Shefali is the director of data-centric AI at Elucidata. She is leading Elucidata's effort to understand the impact of data on outcomes in AI for preclinical R&D. Prior to her current role, she led teams that use biomolecular data to understand disease biology and for drug discovery. She has a PhD and master's from Massachusetts Institute of Technology and an MTech and BTech from Indian Institute of Technology in Delhi. Henry is the Vice President and Head of Machine Learning Research and Cheminformatics at Atomwise. Henry concurrently holds an Associate Adjunct Professor appointment with the Department of Therapeutic Sciences and Bioengineering at the University of California, San Francisco. And he was the 2018-20 Mercator Fellow with the Deutsche, I'm not going to be able to say that one. The DFG, Henry, I'm sorry. That's a big German word. The German equivalent of this U.S. National Science Foundation. Before joining Atomwise, Henry led a research group in computational structure protein dynamics at SLAC National Accelerator Laboratory at Stanford University. His research group was funded by the National Institutes of Health, the U.S. Department of Energy, and by pharmaceutical companies. Henry holds a PhD from the University of Alabama at Birmingham and an MS from Delft University of Technology, both in mathematics. He is the author of more than 150 peer-reviewed scientific publications and numerous open-source scientific software packages. He is a 2019-21 All-American athlete in triathlon and a multiple Ironman World Championship qualifier. All right, let's go down to Felix. Felix is the CEO and founder of Altus Labs, a computational imaging company helping biopharma accelerate and de-risk all phases of clinical development. Felix believes that deep learning models trained on decades of real-world imaging data can be harnessed to better predict clinically meaningful outcomes to sponsors so that sponsors can run smaller, faster, and more successful clinical trials. His team's research, which lever over 140 million proprietary images with associated clinical information, the largest cancer imaging database in the world, has been presented at the ASCO and published in the Journal of Clinical Oncology in collaboration with leading cancer centers. And Felix is a superstar too. He's very humble. He's achieved a lot of things in his life so far. Ali Madani is currently Director of Machine Learning at Cyclica, a Toronto-based biotechnology company. He is the editor of Special Topic Artificial Intelligence in Cancer Diagnosis and Therapy at MDPI. He published multiple papers in high-impact factor journals and prestigious conferences on topics like transfer learning, representation learning, and biomarker discovery. Ali obtained his PhD at the University of Toronto, focusing on application of machine learning in cancer medicine and biomarker discovery after finishing his 2 masters in mathematics and engineering at the University of Waterloo. All right, fantastic. So, highly accomplished people doing really great things for the world. Let's kind of get into the round table. I'll ask the first question. And remember, anyone out there in the audience can ask questions, put them in the chat or the Q&A box, and we'll get to those. So the first question is, we know how difficult drug and therapy discovery are. As machine learning experts in software services companies, describe difficulty you encountered and how were you able to overcome or still address some of these difficulties? Shefali, I'll start with you and I'll go across.
Okay, wonderful. Thank you, John, for that introduction. So yes, I think as you mentioned that the drug and therapy discovery is really complex, and it's becoming more complex because we are using a lot of data, and more importantly, a wide variety of data. So for example, in the same machine learning project, you may be using RNA sequencing data combined with mass spectrometry data combined with flow cytometry data. And you really need to understand the nuances of each of these different types of data, because they're all different formats. You can't just throw all of this data at your machine learning model. And even if you're using same type of data, often if you get gene expression data from one research group or another research group or a different CRO, they will also look different. So the data formats out there are very, very different. And a second, I think the second challenge that we have seen repeated across many, many projects that we've done is that the metadata that are used to find and identify what an experiment was and what is in a sample are not standardized. So to take a very simple example, acute myeloid leukemia may be written as its full term, or it may just be mentioned as AML. And this is across all different types of metadata. And so it becomes very, very difficult to find the relevant data to actually use. And so the way we have tried to address these challenges is that we actually use machine learning, specifically natural language processing based models for metadata curation. And to standardize these metadata for different kinds of biomolecular data. So we have around 1.5 million data sets on our platform, and we have a technology that we can use to curate the data that's already on our platform, as well as proprietary data for our customers. And then on top of this, we do a lot of data engineering to standardize all of these data and make them machine readable, so that they can be read by machines and directly plugged into machine learning models. And add one more thing, I think an aspect that we don't quite talk about a lot, but it's important in every single project that we do, is specifically for applications in biology and drug discovery. There is a lot of computational expertise that goes in, but also a lot of biology or domain expertise that goes in. And it's quite a challenge to kind of have these different folks like data scientists working with biologists or bioinformatics scientists or machine learning engineers working together. I don't think we've quite solved that challenge. We try to create interdisciplinary teams, but that's something you have to kind of worry about for every single project.
Okay, Ali?
Yeah, so I want to partially echo what Shefali said, but I want to get into a little bit more details regarding how we approach the data-related issue, because so technically data is the backbone of every single machine learning modeling pipeline, so technology. So, and the objective that every company has in the business side and the problem that we want to solve determines the challenges that we might have in the data side of things. So for example, if we go back to what we wanted to do in the drug discovery side was to improve hit discovery, to develop a technology that can help our internal drug discovery scientists and our partners and collaborators to do better hit discovery and identify small molecules that can interact with one protein, because usually that is how people want to approach the problem. But we knew in reality that is not what's happening. So we had this idea of polypharmacology, which means one ligand, one small molecule can interact with a bunch of proteins. And the reason, which is very much related to machine learning, is there are some common patterns between proteins. It could be chemical similarity, chemical content similarity, or it could be geometrical similarity between the proteins. So to be able to tackle this challenge, we developed Matchmaker. So which was the technology we developed 4 years ago, and we went through rounds of continuous improvement, and we do that improvement. But let me get into a little bit of details of the challenge that we try to solve. So the data that most companies and we use for drug discovery or clinical trial design is sparse. So in our case, it means that we have proteins with very low number of data points, meaning with very low number of ligands that have been already tested for those proteins, while we have other proteins with higher number of data points we call DTIs, drug target interaction data points. And the question was, if we want to have a generalizable model for the proteins with low number of data points or for proteins with no data points, how could we combine all the data points we had across proteins and even across species to be able to design new drugs, come up with hits for those proteins and develop new drugs for different kind of diseases. So, and that's how Matchmaker works. So we combine all these kind of information to tackle this challenge. So, but it's not necessarily limited. I have other examples for the sake of time. I'm not going to get into the training side, but I will also touch on the testing side, because another challenge that usually happens in machine learning modeling is to make sure the testing strategy is reflective of what's going to happen in reality. For example, in drug discovery side, or for many companies, that is not necessarily in the healthcare setting, it's about customer or client data per se. So, and that is another challenge, because how we define our test sets and how we define and design our testing strategy should be reflective of that. So, and that is possible by collaborating between the expertise in the company.
Got it. Okay, Henry.
Thanks, John, and thanks for having us on this forum. It's really great to be also in the company of so many experts in the field and exchange some dialogue. So I've heard two things really so far. I think Shefali talked a lot about data quality, and that's of course a topic near and dear to our heart, because data drives our models. We have about 17 million data points to drive our discovery models, and so we spent a lot of time curating those data points, of course. And then Ali talked a lot about, in a way, as if I can summarize it, generalizability, which is something that we focus on quite a lot. So Atomwise is a structure-based drug discovery company. So, given a target, we try to identify new chemical matter, given the structure for that therapeutic target. So one of the issues that we have spent a lot of time thinking about is how to make our models more generalizable. And the kind of key to making that work is, no surprise, in the data. One of the issues with the data that we have is, as Ali already mentioned, you have a lot of positive data points of drugs that interact with a target. But we all know that machine learning models also need negative examples. And so a key question here is, how do you come up with realistic counterexamples for your model that doesn't create a simple hyperplane for your model to really realize that, this is a synthetic data point and that's a real data point. And so I'm going to split on that. That's something that I think we spent probably 80% of our time thinking about that particular issue. We augment the data with negative data, and then we have a suite of benchmarks that that ensures us that our models generalize well beyond the test data, as Ali also already kind of alluded to. What we also added is anti-benchmarks. So benchmarks address the generalizability of your model. Anti-benchmarks try to tell you if your model cheats. So it tries to identify hyperplanes. And to give a simple example of an anti-benchmark is, for instance, if you look at all chemical groups that your model is excited about, Is it always, do always certain chemical groups flow to the surface? That's an indication that your model is not really looking at the physical interactions of a ligand and a protein, but it is acting on something else. So in summary, negative examples, benchmarks and anti-benchmarks is a really key driver for our models.
Okay, great. So I think if I used an analogy, a real easy one that people would get, if you've ever painted a room, you've got to do a lot of prep work before you actually paint the room. And I think applying machine learning is the very same thing. So what percentage, just like a number of data preparing the data before you actually start to, build the model and run it. What do you guys think the delineation of time is, so everybody has a firm understanding of like what it takes to actually do this? Am I coherent? Does that make sense? Like, is it 80% data preparedness and then 25% model building and validation, et cetera? So what do you think, Shefali?
I'm not sure if Felix got a chance to.
Well, he's gonna.
Okay,
Felix.
right.
Yeah. Felix is
You're the boss.
Yeah.
Yes, sure. So I think it's. I want to sort of slightly modify the question. In many cases, it's definitely data preparation. I would say is somewhere around 80 to 90% of the time. But part of that is also, I'd split it into data preparation and also finding the relevant data. We work a lot with small biotech and very early stage biotech companies as well. And they don't often have the data available for whatever questions they're trying to answer. So they tend to use a mix of their proprietary data with publicly available data. And so a big part of actually that effort goes in finding and selecting the right data to work with, and then wrangling with that data and preparing the data to feed into models. And that's anywhere easily between definitely over 80%, 80 to 90. Yeah.
Okay, Ollie.
Yeah, so I agree with the 80% kind of range. So it's because anyway, whatever experiment we want to do for continuous improvement in the machine learning life cycle is about preparation of some data and then trying to do any sort of algorithmic changes to see if we can improve our kind of model. Like for the anti-benchmark example that Henry mentioned, so that was a great example. So that needs data preparation definitely for just assessing the preparation of that anti-benchmark and assessing how the model works. But definitely the majority part of the time goes for proper data in the training and tested preparation and featureization. Okay.
Henry, you can kind of concur with that?
I do. It's hard to put a number on it. We also have an entire team devoted to developing new architectures for machine learning models. So beyond what is published in the literature, simply because we want to be a leader in this field. They obviously, it's a team of about 5 people. They obviously spend 100% of their time developing new architectures. When a new model is about to release, of course, we shift towards preparing the data. And within Atomwise, we release new data cycles on a specific cadence, and we have a team dedicated full-time to that as well. So it's a major effort for us.
Okay, so Felix, so now I can kind of get to you because you're dealing with clinical data many times, translational data. You're dealing with, hopefully standards, but not always. The metadata, the labeling, the tagging. Why don't you just kind of describe the level of effort that you go through, even though you're a little further downstream? You're, it's a very comparable thing, I think.
Yeah, that's absolutely right. Data prep is similar to everyone else here, probably over 90% of the work that we do, and because we're pulling in different types of data. So our models use the imaging data as inputs, radiological imaging data, but in addition to the imaging data, we combine that with long-term claims data, outcomes, treatment history. We combine that with data from lab information systems to get the histology and any sort of molecular testing that was done for these individual patients. We combine it with functional testing, pulmonary function testing, other types of testing that's been done, all the way through to adverse events and toxicity information. So all of that takes a lot of curation, a lot of structuring, and getting the time points right, understanding the chronological order of imaging, of events like treatment start, outcomes, and so forth. And that is really 90% of the work that then also deals with normalizing that data, pre-processing that data so that the models that we generate from that are generalizable.
Okay, and because it's imaging, you actually can start to see adjacencies. Maybe sometimes you can't see in small molecule work, but I'll kind of ask that. So you can actually start to see comorbidities and other things like that because, picture's worth 1,000 words, right? You're starting to see things around that and can bring that into your environment as well. So that I thought that was pretty cool.
Absolutely, yeah. I mean, when you think about a CT scan of a patient, it's their entire body.
Yeah.
So our models are not just looking at tumors, which are, what currently is measured in cancer trials. Our models are looking at all the tissue within the body and can therefore, when our models are predicting outcomes, pick up on, cardiovascular diseases that a lung cancer patient might have, or body composition, which we know has a lot of prognostic value, age from the density of the bones, sex, histology, things that we humans can't quantify from the imaging data, but that from seeing millions of examples, our models are actually able to pick up on, which is super fascinating.
Yeah, very cool. All right, so I just wanted to touch on one thing. So what everybody just described was a lot of the data, right? And I think this goes back to the hype piece, like companies all of a sudden ran after machine learning and did some of these things without really having model quality data and enough of that data in their environments. And so maybe just ask this quick question: how has that impacted your company, right? So I see, and this is another correction too, you either agree or disagree. I see sometimes the big companies throw into really difficult problems at some of the partner vendors. Like, wow, we'll just give this to them. We couldn't solve it for the past 10 years. And it's really difficult. And they're handling all the day-to-day stuff. But then, obviously you have smaller clients and stuff too. So kind of just talk about how the hype has either helped or hurt you, and then throw back in some of that data, that the data observations, and how it's like dealing with larger versus smaller clients, etc. Because we have a mix of clients on here that are small clients and our big clients, and so kind of just elaborate on that a little bit. And then Henry, I'll start with you and we'll go back the other way.
So yeah, how has the hype helped us? Well, it definitely helped in fundraising.
Yes,
that's for sure.
That's actually a very important point. If you had AI ML in part of your strategy in your company, that helped a lot of companies get money. So that's a really good point.
Yeah. So I think so. Atomwise started out off, initially as a platform company. During our Series B, about a year and a half ago, we made a hard pivot to being a pharmaceutical company. And so we are no longer sort of really exposed to the kind of hype that you described, like working with partners that then throw out in a way a bone and it's up to you to solve it. We're in it for the long run, and so we have a portfolio of about 20 therapeutic targets that we work on with our own technology. And the way I view, so what Atomwise is trying to do is to turn the drug discovery process from sort of a human-driven kind of idiosyncratic process into really drug engineering. And of course, that's a much longer-term process than a quick win using AI. And in that sense, it's more like a marathon for us than a sprint. And we're obviously in a biotech downturn at this point. And I think what that will help do is sort of shake out some of the hype. We'll see, some companies that have tried to capitalize on this technology only without having a broader vision on how AI sort of enables us to do things that were previously difficulty, but at the other side, we still need sort of the human insight to drive this discovery process forward. And I think once we get there, we will see that, we'll get there eventually with the help of machine learning.
Okay. Ollie?
Yeah, so that's a good and tricky question. Definitely it has been helpful in the fundraising side. But one thing which I'm very grateful of the senior management coming from CEO, and so the plan and also the kind of culture was to not overpromise, never. So, and that is very important in the area of when we have hype in AI and ML being a magic bullet and solving everything. So internally, through our internal drug discovery program, we have drugs and therapeutics in our internal drug discovery program, or previously or currently with our partners. We just try to make sure we are informing everyone with full honesty if there is any sort of bias in our platforms, in our technologies, and what is possible and what would be the likelihood of success in every program. So I think that could help in long run the whole community to just make sure that hype doesn't hurt anyone in the user level or in the development level.
Okay. Shefali?
Yes, that's a very interesting question. So I think for us, around 2015 is when Elucidata started, and that was also the time when every startup was trying to use AI or ML for their technology. So we really had to actually resist using that in fundraising in our initial years. Because at that time, we were not using machine learning for data curation. That was something that emerged after the work that we had done with a lot of customers to really identify what are the core data problems that our customers were facing. So we in some ways we have to push back. We had to push back on the hype and try to throw in the word ML during fundraising until we had a product that is now heavily supported by. We use NLP in our metadata curation efforts to curate data at scale. So we had to push back initially and be very, very mindful and deliberate about when to start using machine learning for fundraising. And of course, and we had to wait to identify that problem and build that technology to support our product to start using that. And I think what really helped us do that is this services side of the company. So we still, even though we are a tech company and a product company, we still work closely with our customers one-on-one. And so those interactions really keep our focus on the problem that our customers are trying to solve. The technology or AI is not the focus. The focus is what comes out of it. And that sort of feedback, I think, keeps us really on our toes and mindful not to get too drawn into the hype.
Right.
I would say. Yeah.
Okay. So Felix, you again, you're a little different, but the imaging data is based on something that, in many cases, not all, you can see, right? So you're taking a lot of that data and stuff. So what are your thoughts around this? I mean, so I don't think a lot of what you at Altus did was hype. It was based on solid imaging data, et cetera. But what are your thoughts?
Yeah, I think AI applied to imaging has probably gone through 3 hype cycles now over the past since 2014. So 8 years, and it's been interesting to see investor interest sort of increase and wane through those cycles. It's at the end of the day, it's about serving our customers. And for us, we're 3 years old now. I think Ali mentioned this previously. It's about delivering on your promises, and over promising can really be hurtful to companies, as we've seen in the imaging space. And with IBM Watson, for example, right? Some of the promises that they made in the applications of AI in medicine. And I think it's really important to be honest, to ensure high data quality, to publish results, and really be diligent about the claims that are being made there. It's nice to get investor interest, but at the end of the day, it's really about serving your customers, and being consistent in the quality of your products that you're providing to them.
Okay.
And of course, too, if I may, John, I think these
please.
are excellent. These are excellent points, and I think what's important, of course, for all of us, like ultimately the proof is in the pudding, right? I think we make predictions with our models. All of us send out the data to in our case CROs, and so if our model predicts hits to a specific targets, we 4 weeks later, we get the data back and we know how our models perform. And not once, we do this, on a daily basis. And so that helps us keeping track of the performance of our AI models, like almost in real time. And so that will overcome the hype. I think, ultimately nobody, at least in drug discovery, nobody cares how you got a cancer drug, right?
Yeah.
And if AI can deliver on that promise, it's that's ultimately the goal, I think.
Okay. Yeah, and I think there's a whole probably another conversation here. I remember in my own experiences years ago, just the semantics of predictive versus suggestive, and the suggestive was a much better way to work with a medicinal synthetic chemist, because you weren't telling them what to do. You were kind of guiding them a little bit and giving them more knowledge and information. And that semantic change made a huge difference in some of the work that I did. So it's a very interesting dynamic. Go ahead, Shefali. Yeah, please.
So what you said about suggestive and predictive, actually, we've observed the same thing with our data curation model. So to be able to build these NLP models for very specific curation, metadata curation tasks, we do have to generate some, we do have to generate some training data, and we have curators who help us do that. And we've noticed sort of a huge difference in the quality of those labels that our curators do with model-assisted curation. So we provide them suggestions weekly annotated with our previous models, and that has led to a huge jump in label quality.
Okay. Anybody else want to comment on that before we go on to another question? Okay. Technology. The audience is going to want to know what type of technologies are you using to curate, for model building, model validation. Like, just kind of go through that. Obviously not giving away any proprietary things, but in general, like how is that process, and what type of technologies are you using to accomplish that? Shefali, you can go first.
I'm not sure how much detail I can go into.
Or maybe even how you use it, how you use it, or what you use, just say obviously what you could.
Sure, I can talk about a few aspects of it.
Yeah.
So as I mentioned earlier, that we use NLP-based models to curate metadata at scale. And then we have several data pipelines, data engineering pipelines, basically to structure the data alongside curating the metadata. So in our metadata curation process, we definitely have, so the way we've designed that process is kind of to really get high quality labeling data. So for example, we've built sort of double blinded label curation in our training data generation. So every label goes to at least 2 curators, and there's consensus building, and that's how we kind of ensure high data quality for that particular task. And for a lot of, I think the rest of the technology stack, once the data are curated, structured, and they are on our platform. I think that's proprietary.
Okay. Yeah. Okay, Ollie.
Yeah, so part of that goes to just Python, PyTorch for deep learning, and for example, traditional scikit-learn for machine learning. And I think there are many blog posts for that, but I can get into a little bit of how we work. So depending on the type of data and objective, that determines what kind of technology we can use, right? So for example, if we can use graph convolutional network that we use, or multi-layer perceptron that we use, or different kind of machine learning modeling, similarity-based, rank-based, all this kind of thing to achieve those objectives. One part of how we organize the teams in the platform department that we have is to benefit from different technologies. For example, in the protein structure side and pocket identification, and then in the drug target interaction prediction, and considering different expertise different experts focus on those specific problems, then we could come up with this scalable kind of system just to increase our quality in the protein side, our quality in the drug target interaction side. But the technology side is very common. It's all usually based on what at least we know is Python, PyTorch, TensorFlow, still in many companies. And the rest is just a matter of if there is a need for any public tool to be used in the drug discovery side that we haven't developed it ourselves. If there's a private tool that we have to license to use and we haven't developed it ourselves, if there's a potential for new technology development for data integration or prediction. So these are different kind of things that need to be considered.
Okay, Henry, you're up.
Yeah, in a way, what Ali says. That's I think, you know, we use this, like many of our competitors. We use the standard tech stack to build our models. You know, one thing I think that Ali also touched upon is that in deploying these models, I think that technology is probably less important than creating the right teams to build these models. And so what we try to do is really lean heavily on domain knowledge, because you know, AI is just a technique that helps, you know, domain experts to reach their best potential. And so what that allows us to do, for example, is I'll just name two examples. So one is in the later stages of drug discovery, you know, when you are in lead optimization and you try to optimize other properties of the molecule than just binding to the target, data gets really scarce. You're talking about, you know, hundreds of endpoints for a particular target, but there are about, you know, roughly 25 endpoints that you want to optimize simultaneously. And so one of the ways that you can build on domain knowledge is realizing that many of these endpoints are correlated. So you start thinking about building multitask models to capitalize on these on these correlations, or, you know, pre-train your model. Pre-training, of course, is now a big thing in machine learning. You have these big pre-trained models that you then fine-tune on a particular subset of your data. And again, this requires a lot of domain knowledge. And that's very closely related to the second example that I was going to mention, that is also really, you know, has come up in the last few years, and that's active learning, where, you know, you have these rapid cycles of prediction. You get data back from your CRO, you retrain the model, taking the new data into account, and you go through a next cycle. And that, I think, is where, you know, that ties back to something you suggested, John, where, you know, instead of telling your medicinal chemists, okay, here's the silver bullet, just make this molecule and, you know, we'll be in business. It's like, okay, we have, you know, we generate 100 hypotheses for you that, you know, populate your SAR table. And now you send it out to a CRO, you get your data back, and we go through the next cycle of hypotheses. So in a nutshell, I think domain knowledge is really what makes a lot of the things work for us.
So it was actually, you're one step ahead of me, Henry. So it was going to kind of be part of my next question. Is it fair to say a lot of the companies that started out maybe 10 years ago and said, we don't have domain knowledge, we're mathematicians, we're going to apply these models, they've kind of faded away a little bit. I think it's this, that the companies that have the intense domain knowledge and the intense machine learning experience or capabilities that are still here today and actually making a difference. Since you were last, Henry, I'll just ask you if you agree with that or disagree, and we'll just go around real quick to see what everyone has to say.
Well, since you mentioned mathematicians, I'm on the side of I'm in the camp that, ultimately, we hope to get beyond domain knowledge. We hope to transfer that kind of domain knowledge to our models. In reality, we are a long way away from that. So I do tend to agree with you, John. Yeah.
Okay. Ollie, Shafali, Felix, you guys can chime in on that.
Yeah, I definitely agree, John. Even if they haven't faded away, they learn their lessons that maybe it's the domain knowledge need to be incorporated. I have a mathematics background from master's, but I had to learn many things to get to here. And also, sorry, going back to what Henry mentioned, it is about collaboration within the company. So we have protein scientists and the chemoinformatician and other kind of expertise, and we inform each other for any sort of planning and decision-making and technology development. That matters a lot. Nobody can be A+ like in a school for all courses.
Right. Shafali?
Yeah, very, very strongly agree with what Henry and Ali said. We actually started with domain experts. We spend a lot of time actually doing these machine learning projects, working with the types of data that we work with our customers, before we actually then start bringing machine learning experts into the team. And that was really critical. We were, I can't even imagine how we could have done it the other way around.
Right.
So yeah, domain knowledge is extremely critical, and I think another place where I very strongly agree with Felix, I would love to see a future where that domain knowledge can be integrated into our machine learning models, but we're definitely not there yet. But there is a lot of good research happening on how that can happen. Right now, domain experts are really the only way to integrate that and bring that knowledge to the models through our data.
Yeah. I think it's happening. So years ago, talking to Chris Lipinski, he said, hey, it takes about 10 years for a chemist to hit their stride. And ever since he told me that, one of my goals was, how could I shorten that? How could I get that down to 7 years or 6 years, right? And machine learning and computational sciences and stuff, using those methodologies and that in silico first approach, educating people more and more and more, getting them to hit their stride earlier has always been a goal that I've had. But I think we're getting there, definitely. Felix, you're working with MDs, DOs, clinicians and stuff. So it's slightly different, but what are your thoughts?
Yeah, very much agree. The domain expertise is crucial, as is the machine learning expertise. That being said, just the third really important component there is that the access to large scale and high quality data as the third component. That is also crucial here. On the ML side, we've seen how fast that research has evolved and continues to accelerate in terms of new papers, new techniques, new architectures being published. So I think that will become more and more a commodity. Whereas it's really the domain expertise and the proprietary access to data that will continue to differentiate going forward.
Okay. Let me get to some audience questions. Typically, what are the broad technologies that can be used for integrating different types of disparate data? So this is kind of a scientific data management question. Maybe just quick go through and just let's give a quick answer on this from each of you. Shafali, why don't you start?
Sure. Yeah, that's kind of a broad question. So
It is.
it depends on the problem that you're trying. Your data integration strategy will depend on the problem you're trying to solve. Having said that, we chose to solve one part of it, of the problem, which was metadata curation. So metadata is common across all different types of data that we use, and so we started with the problem of metadata curation. And then downstream, when you actually take those data and then use it for a particular application, then there are several other, I'm sure, techniques that are important, especially with biology. You have to worry about whether your data is truly showing biological variation or technical variation, and then you have to correct for those artifacts, if that's the case. But that gets specific to the type of problem being solved.
Yeah. Does anybody else want to touch on this? I can say a couple things if you guys want to kind of, it's up to you. Chime in now if you want to. All right. So, you know, we have all, all types of technology here, data lakes, but it's actually what Shefali's saying. If your data isn't contextualized, if you have garbage in, you're going to get garbage out. So it's a process here to bring that all together. And you could potentially use machine learning to do some of that, but my experience has shown that 80% of legacy data is missing enough contextualization where there's a serious decision on whether you include or exclude that data in your bringing together of that data. Let's go to the next question. Where do you see the machine learning technology and biomolecular research 10 to 15 years down the line? Henry, I'll start with you on that one. Future forward-looking statement.
Sorry, can you repeat the
10 to
question?
15 years. Where do you see machine learning 10 to 15 years from now in biomolecular applications?
Let me look at my crystal ball here. So there is no doubt that I think in the kind of work that we do, like the drug discovery pipeline from hit ID all the way to lead optimization, machine learning will radically change the way we think about that. Will it be completely automated 10, 15 years from now? Probably not. But will it be largely automated, and will AI play a major role in creating these kind of hypotheses as we just discussed? I have no doubt about that. So the future of drug discovery and machine learning really, I think, is commoditization of the drug discovery process.
Okay, Ali.
Yeah, so as Henry said, the initial stage of drug discovery definitely is under development. That's been happening sooner or later. One thing, it's not that I know or I just expect it happens, also part of a wish. I hope we somehow be able to tackle translatability gap, because whatever we do in earlier stages of drug discovery, many of them fails in clinical trials, and majority of the cost of drug development is in the clinical trial. And I hope I think we will get there with the amount of data that is getting generated to fill that gap and be able to decrease the cost and make the process more successful.
Felix, Felix, go ahead.
Yeah, that's a great point, Ali. I just wanted to build off of that and mention that, I think within 15 years' time, we hope that, every phase 3 trial will be successful. And because we hope that in phase 1 and 2, we'll be able to create synthetic control data, from single arm trials that will give us enough confidence to run a randomized controlled trial that will always be successful. That is where I hope we'll be in 15 years' time. And really the combination of different data modalities that are already being collected in those clinical trials, from imaging plus, blood samples, tissue samples, various types of omics. I think that is where we're going to see a lot of exciting developments in the next decade and a half.
Yep. Shefali?
I would just add that I don't know what's going to happen, but I hope that we see, just like in the last 10 years, we've seen research on machine learning models and commoditizing those models, as Henry said, really advance the state of the field. I hope we see similar investments into data quality improvements and in systematically improving data quality and figuring out how to do that. I seriously hope that that's what we'll see.
Yeah, we have a cultural problem, unfortunately, with data, and it starts very early. And until the culture changes in a lot of these organizations, truly changes, we're not going to see an improvement in model quality data and enough of it and stuff. So obviously companies are, supporting the FAIR principles and doing this and that, but there's still not enough there. And I work in it every day. So until we get this culture right, from the time of design of experiment, data capture, and all the way out to reuse, that has to change. And it's changing, but slowly. So that's I think our big problem. We have 2 questions. We're we're at the end, but can you guys stay on for another 5 minutes, 10 minutes? Does anybody have to run? Henry?
I need to drop off.
Okay. All right.
Feel free to continue without me, of course.
That's unfortunate. Thank you so much, Henry. It's been great. We really appreciate your insightfulness and your answers. Thank you so much.
Thanks again for having me. Take care.
Okay. So one question is around the lack of reproducibility we're seeing in clinical and peer-reviewed. This is a huge problem. This is part of this culture that we're talking about. And the question is around, having more robotic cloud labs, where things are very reproducible, very repeatable. I definitely think that will start to drive this in the near future as well. If anybody wants to comment on that, you can. I just wanted to get to this next question about how about deep reinforcement learning in biomolecular applications? Go ahead, Ali. You're smiling the most.
I can touch a little bit on that because we had that conversation a couple of weeks ago with a friend of mine. So deep reinforcement learning in the off policy, that is the ultimate kind of goal that many people are excited about, is not that successful right now in many fields. And the reason is data efficiency. So deep reinforcement learning in general is not a deep data efficient kind of modeling right now, and definitely it's going to change in the near future, and that limits its application, in my opinion, for biomolecular research in general, because we have data limitation issue all the time regarding, for example, proteins, patients, or so. But I would be happy to continue this conversation through LinkedIn or email. So I love the idea of applying deep reinforcement learning in our domains.
Okay, Shefali and Felix, you want to touch on that? Add to anything there? Nope.
Not really relevant.
Okay.
To the work that we do.
Some comments about, this will be the last question. Some comments about multi-target validation would be great. So I know Ali touched on polypharmacology a little bit earlier. Shefali, anything come to mind from an elucidated perspective where you might be able to touch on that? With Felix, we talked about those comorbidities that are in and around what they might be looking at, but this one might be more for Shefali and Ali.
I am not quite sure, actually. I'm not quite sure how to answer that.
I'm not 100% sure if he's talking about more like a polypharmacology approach, Ali, or is this more of a machine learning multi-target, right? Finding the right target for the disease. So this is more like,
okay.
Oh yeah, okay. So this is more
That's
target discovery kind of question.
That's
exactly.
$1 billion question.
Yes.
If you can figure out how to do that.
Right.
No, I'll briefly comment that's definitely a very hard question. I think at the heart of it is how to use machine learning to do that well. And part of the problem that I mentioned is often a problem like target discovery involves an interaction between many different biomolecular and molecular entities at the cellular level. And so you're working with, for target prediction from a biomolecular data perspective, you're working with many different kinds of data. So you're working with often gene expression data or DNA sequencing data or proteomics and metabolomics and all of that together. So it's really about, for a particular problem, finding those right data, understanding the nuances of those data, cleaning them up, which is where most of the work is, and then try to model, disease biology. So again, data sort of is also central to this problem, and domain, and not just availability of that data, but sort of understanding of that data.
If you have a minute, John, I can add a little bit to that. So I think one part of the problem is more of a comment, not an answer, not a solution. So, but one part of the problem is heterogeneity in different kinds of diseases like cancer, which goes back to the data center kind of approach. Because if we have a more kind of homogeneous data, we can come up with a cleaner target and more kind of target identification for different subpopulation of patients in different diseases like breast cancer, pancreatic cancer, or other kind of cancer types, or even other diseases. So that's definitely one part of the challenge. And single cell data, I think the premise of that was to help in purifying these kind of target identification or biology kind of identification, and it's been partially successful.
Yeah, and I can comment on this a little bit too, because I work with several clients, large pharma clients, where target ID and validation is actually a microcosm of what goes on after all that as well. So it's very, very important to stay extremely organized and almost on the same scientific informatics or footprint of tools and things that you use early on, so that you have this ability to collaborate and share along the whole drug discovery process. It can't be like a question before about disparate data. Oh, in target ID, we're just going to use all of our own tools and methods, and then we'll throw it over the fence to the drug discovery teams. It's not efficient to do it that way. It's efficient to be highly collaborative, using in the same environments and being able to share data, because again, I'll just emphasize it one more time. This is an extremely difficult process, drug and therapy discovery. And you need to be on your game for communication, for collaborating, for understanding the science, the domain experience, the technology, and I just go down the line. So the more that's collaborative, the better and the more successful you're going to be, in my opinion, from what I've experienced over the past 30 years. Felix, any comments on that? It's kind of more target ID, and you're kind of more at the other end, right?
Yeah, but I think you make a great point that when you're defining a target, you really need to, I guess, think about the likelihood that target is actually going to be meaningful, right downstream. How likely is that going to result in positive,
right,
preclinical results, ideally clinical results too. I mean, every drug that makes it into phase 1 has biological plausibility, past the preclinical stage. And then, of all the phase 1 indications, you got less than 10% that make it to approval. And of those, only half actually have evidence of clinical benefit. And we only understand the mechanism of action for drugs that work, of only a portion of those. So it's a lot of guessing. So I think it's a tricky space, and we're lucky to be in the clinical development space, where our ground truth is survival as a clinically meaningful outcome. Yeah.
Okay, awesome. Well, I want to thank everybody the audience. I want to thank who's left after Henry had to leave. So Shafali, Ali, Felix, this has been great. I think what it gave is an element of truth to the audience of like what you have to do to deploy or implement machine learning. It's like I said, it's like painting a room, right? You got to scrape, you got to do all this prep work to actually get to the model part, which is, you know, where then the real things start to happen. But if we can change our culture and start to view data and the processes that produce the data as a bigger asset than they're really looked at today, we'll get there before 10 or 15 years. We'll start to really have a paradigm change when it comes to this. And unfortunately, I knew this back in 2000 when I was doing a lot of this work, but not much, a little has changed since then. So I think if we just keep going, we'll get there. That's what I'm saying. So thank you very much, and have a great rest of your week.
Thanks for hosting, John.
Thank
All
you,
right.
Jen.
Yep.
That's
Take care.
great.
Thanks,
Thank you.
everyone.
Appreciate
Bye.
it. Bye.